chemistry in homeostasis. So, okay. Always autoimmune disease, it is a multi-factor disease. So genes and immune regulation and environment, they all together take a part of development of autoimmune disease. And genetics factors specifically, these genes are related to immunoglobulins, T cells, receptor, the major histocompatibility complex. The first two, which are involved in the recognition of antigens, are inherently variable and suspicious to recombination. This variation enables the immune system to respond to a very wide variation of individuals, but may also give rise to lymphocytes capable of self-reactivity. So it's some single genes models that can cause autoimmune disease. Some of this you know, but this is mainly when. So, all genomic studies need more data for create more accuracy practice and fundamental knowledge. And for achieve this goal, we need right tools. One of these tools can be a system of biobank, because we need really a lot of data to achieve specific research. So, biobanks collect biological samples and associated data for medical scientific research and diagnostic purpose, and originates this in a systematic way for use by our. The collation of samples and data for research purpose has a long history in the education and medical system. In the past, biorepositoriums were relatively uncontroversial, residing largely in the seclusion of pathological institute. With a recent technology advance, the potential to open this exciting collection for new use is starting to be realized, but also new biobanks are being established. Innovation in informatic technology enable the systematic collection, linkage, and tracking of the sample and data, but also provide the tools for analysis across vast sample and data sets. What distinguishes present from the past is the general scientific context has changed, and the scale of biobank achievement both in terms of quantity of samples and data, as well as the range of disease area and institution now involved in biobanking can increase considerably. Where our significant can change is that this collection are bioconquerors, so that can be used as a resource for whole scientific community. So, as a result, biobanks are useful tools for reduce the time and expense of research, should be set up or collaborated way where infrastructure is shared. Request robots, legal and ethical regulation which idea of regulation hubs should be encouraged. Why do we need a biobank for studying of autoimmune disease? A biobank for integration of molecular data with the clinical information represents a single organized common resource that can be utilized to perform multiple hypothesis-driven studies, interface with industry, and monitor the evolution of autoimmune disease in our society. Advancing in bioinformatics and computing technology has enabled scientific research to be organized and carried out in new ways. Scientific practice can become increasingly interdisciplinary with a rapid formation of flexibility and dynamic research collaboration around the world. Research projects are frankly global in nature, involving teams with different types of expertise, such as clinical, laboratory staff, and research, bioinformatics, statistics, and our data analysis. Biobanks are embedded in this complex network of research collaboration that span regions, countries, and the globe. As a result, there are many different types of biobanks that have been built for a range of different purposes and reasons. The longer-term vision for biobanks with Europe and America is to facilitate the linkage of the different biobanks as a part of meta-leo infrastructure across all globe. The expanded pegs on the medical research drug development and healthcare are anticipated to significant. As such, infrastructure will enable new scientific questions to be rapidly addressed when the crisis efficiently. But this is a problem with biobanks. Most of people have never heard about biobanks, and many citizens appear to be reluctant to participate in biobank research as a donor to biobanks or a participant in cohort study. At the same time, there are concerns among citizens that eventually biobank research might turn against them for violation of a privacy right or being disadvantaged to them terms of insurance or employee discrimination. In addition, the legal and regulatory frameworks that apply to this area are fragmented, with variation of practices across different areas of medical research. Biobank tests our regulatory frameworks and the principle for medical research, but also raise a number of complex issues for society. Thus, we hold that the development of biobank research is not only a matter of scientific excellence and investment and the building of much needed infrastructure. So, advantages of biobank. It's main of it. It's statistical power. Larger number of samples collected over years, but available instantly. So, if I start research in my country, we have not many biobanks, but with network of biobank, I can get access for information what I need. So my research can be more specific, and I have a data that I can analyze by myself. It's very great. So if many scientific can get to this biobank, it's a reduced cost as once collection remains available for a number of studies. Harmonization, standardization, operating procedures, and best practices yields are more efficient and a time-safe collection of a sample which are quality assured. Logistic. A great potential for a translation research project and will facilitate clinical trials. Development of synchronization IT infrastructure creates a case for many more users effective to collaborate from any country, and so it really can unite medical world. Ethics. It's some problem with ethics because single ethical proof and well-controlled ethical process lessens the time in setting up individual biobank as well strengthens client trust. Biobank is a real power because we can analyze our whole population, and if we do it, so we think what some be stronger and some can be less chance for survive in a modern world, because it's a great problem that employers, if he knows that his potential worker can have a deadly ill, he can reject him for work, and it's really scare some people that they cannot access to the dream job because he or she have some genes with them. They statistically can do work what they really like. So, population. Two larger groups of biobanks, whole population and subset of population. Whole population, collection of a sample from a large number of general population, no disease specific, useful for epidemiological studies as well as prospect studies. This type is subject of a genetic passportization. Idea of genetic passportization, it's create some system for healthcare when children, when we born already have some protocols for their future disease or not, if they are absolutely healthy in their genes or some omics study. Subset of population, it's a collection from subset of population with autoimmunity disease, including a family collection useful to identify the cause both genetic and environmental of condition. This type is most interesting for researching. Today, autoimmune disease case of autoimmune disease increase every year, so we need more data for create some protocol of predictive or treatment more specifically by some drugs target. Yeah, it's a kind of picture of that working. So I think I literally finish. Yes, that's all. Yeah.